Bacterial growth curve. In this video, you will learn what a bacterial growth curve is and the factors affecting the growth of microbes in foods. When we talk about microbial growth, we are referring to the number of cells, not the size of the cells. Despite the fact that bacterial cells nearly double in size during their life, it is the number of cells that is most impactful. Microbial populations, or the number of cells present, can become incredibly large in a short period of time. If we understand microbial growth, we can better control the growth of microbes that cause disease or food spoilage. Bacterial communities go through a series of consecutive growth phases. Lag phase. The lag phase is an intense period of metabolic activity where organisms prepare to begin division that leads to population growth. This phase may last from several minutes to several days depending on the type of organism and the factors that affect growth, such as nutrient availability, pH, and moisture. During this phase, we observe little or no cell division, and thus no increase in the number of microbes. Log phase. The log phase, also known as the exponential phase, means that there will be a steady increase in the total number of cells as the organisms rapidly divide. This division is called binary division in that one bacterial cell divides into two. These two produce 4, then 8, and so on. This type of increase is referred to as exponential growth. The population size will continue to increase until it reaches a maximum concentration when growth slows due to the exhaustion of nutrients, accumulation of waste products, and harmful changes in pH, among other factors. Stationary phase. The stationary phase is a period of equilibrium because the number of microbial deaths matches the number of new cells produced and the population size stabilizes. Death phase. During the death phase, the population decreases as the number of cells that die exceeds the number of new cells formed. This phase continues until the population decreases to a small proportion of cells that were present during the stationary phase. However, the death phase may not lead to complete elimination in that some proportion of cells may survive. Water activity, pH, temperature, and nutrient content are the primary factors that influence microbial growth the most. The microflora, or organisms present in a food, is rarely a single culture, that is, one species of microorganism. Rather, foods are often comprised by many different species of microorganisms that originate at some point during the flow of food: growing, harvesting, slaughtering, processing, and storage. Several factors affect microbial growth, which in turn will determine the nature of spoilage or the risk of a health issue. These factors can be summarized in 4 categories: intrinsic, extrinsic, implicit, and processing. The intrinsic factors are the physiochemical properties of the food itself, including nutrient content, water activity, pH value, and the existence of an antimicrobial component. The extrinsic factors include the environmental conditions in which the food is stored, such as relative humidity, temperature, and gaseous atmosphere. The implicit factors are the properties particular to each microbial group, including the interactions among microbes and their interaction with the food, as well as their ability to use certain nutrients, tolerate adverse environmental conditions, and produce antimicrobials. Processing factors are the treatments used in food production, including but not limited to slicing, washing, packing, irradiation, pasteurization, and others. These factors will affect the composition of the food and consequently modify the types and numbers of microorganisms that remain in the food after treatment. Each of the factors mentioned plays an important role in bacterial growth. However, the interactions among them ultimately determine which microorganisms will multiply and which won't. This is the end of our video on bacterial growth and the factors affecting the development of microbes in food. This video was developed by Food Safety CTS, LLC for Penn State University.